Dr. Christopher Shaw is a professor in the Department of Ophthalmology and Visual Sciences at the University of British Columbia and holds cross appointments with the Department of Experimental Medicine and the graduate program in Neuroscience. He is speaking about aluminum as a neurotoxin, the evidence from cell culture, in vivo, and human studies.
Okay, so what I want to talk about today, it's going to be sort of a broad spectrum talk. Let me start by declaring a conflict of interest, as I think we all should in all circumstances. I am the founder and used to be an officer of a early stage biotech company called Neurodyn. Its mandate is to search for early biomarkers of neurological diseases such as Alzheimer's, Parkinson's, and Lou Gehrig's. It has nothing to do with vaccination or vaccination therapy, so we get that out of the way right away. I'm going to talk to you, and again, if anyone wants to see the website, you can go verify it for yourself to see that there is no conflict. The neurological disease clusters is something I want to touch upon very briefly. I want to go into the epidemiology of ALS because that really is what I am. I'm not a vaccine researcher, as I've said several times. I'm an ALS researcher. And we're going to follow a particular disease cluster because it is relevant to this story, I think, called ALS-PDC. It's a big word that or a short acronym for a very, very big word called ALS, which stands for amyotrophic lateral sclerosis, Parkinsonism dementia complex. And we'll talk about what that is, show the links to Gulf War syndrome and where aluminum might come in, and some of the stuff we've done, aluminum neurotoxicity in various preparations that we've looked at, and how we can actually mimic aspects of motor neuron disease, and we think other things with the aluminum adjuvants we've looked at and dietary aluminum, and some very brief conclusions in future directions, where we're going to go from here. So when you want to evaluate a neurological disease, you really have to know some key things. There are things you absolutely need to know, and we often don't. So we want to really know what are the causal events, what's starting the disease process, how does it begin, and really distinguish those from what we call coincident events. When you look at, when one goes to neurological disease conferences, someone will get up and present some data on what's called proteomics or metabolomics or genomics, and you see this wave of colors on the screen. And what they basically are showing you is that lots of things have changed by the time someone comes to clinical diagnosis, or even more so by the time they are dead, and you have bits of their nervous system to look at. Thousands of things are different. And you don't know from that stage what the events that started that cascade of neurological degenerative things might be versus things that are bystander events that just happened. And in fact, as someone once pointed out to me, some of them might be compensatory. Your system, the nervous system is plastic. It's trying to fix itself. It's not just that it's dying and nothing else is happening. It's not like whack-a-mole, one thing is happening and that one bad thing keeps happening. It turns on cascades of events that by themselves can be equally pathological to whatever is initiating it. So many of the things may be compensatory. And I'll tell some people if they want to hear the story of how I went down that path and made that mistake some years ago. So if you know what causes it, you can in principle go to some sort of strategies for prophylaxis. If you know the stages of the disease, how it emerges, and you don't know it from human clinical data because, again, people present to their neurologist with a disease and it's already an established disease. But if you did know that, you could come up with early treatment strategies. And what is a cure? How would you take people back to the point they were before? And that applies whether you're talking about Alzheimer's disease or autism spectrum disorders. And again, there's really no consensus what this means. Does cure mean you go back and you're like you were before anything happened? Or does cure mean that you simply don't get worse? And that's something that is actively debated by neurologists. So one way to think about it is imagine your nervous system at birth is completely healthy. You have all the neurons in perfect condition, and somewhere along there something happens. That something triggers, whether it's a genetic defect or whether it's something environmental, it triggers something that now leads to this cascade of events. So you start here with 100% of all your neurons being happy, and somewhere down there you hit that threshold. And remember, during this period of time, you are apparently normal. You apparently have no neurological disease consequences until you hit that critical threshold of loss, whether it's cells in the substantia nigra in Parkinson's disease, whether it's motor neurons in your spinal cord in ALS, whether it's cells in the hippocampus and cortex in Alzheimer's disease. You hit that critical threshold of loss, and now you present with the symptoms of the disease. And yet, there has been this huge period of time, and we don't know how long that period of time is, in which things were going wrong. Okay? So if you really wanted to prevent the disease, you'd have to be out there. If you wanted early phase treatment, you'd have to be in there. And in fact, where we're stuck for most neurological disease treatment, no matter what that disease is we're stuck down there. And that's a bad place to be. So that's where prevention would be, that's where intervention would be, and of course, the reason neurological disease treatments rarely work, at least in my world of ALS, Parkinson's, Alzheimer's, is we're stuck at a place where so many neurons have been lost, so much destruction has happened, there is nothing you can imagine. There's no drug cocktail. Remember, thousands of things are wrong. What drug cocktail are you going to invent that is going to not only stop those thousands of things, but reverse the damage and now repair neurons? Those are whole different categories of problems and largely at this point intractable problems. So one way to think about causality is to do this kind of Venn diagram that you see increasingly at various conferences. The Venn diagram that actually describes the interaction between environment, genetics, and age. And again, this crops up again in my world a lot, and people have speculated about that, and it's coming into the discussion about things like Alzheimer's. So, for example, the environment clearly can play a role, as can genetics, and where those two intersect is your disease phenotype can become apparent. One of my colleagues in Parkinson's disease often says, and what I should stress, by the way, is that 90+% of the age-related neurological diseases, ALS, Parkinson's, Alzheimer's, are not of genetic origin in the sense that you cannot find a dominant mutant gene of any kind. You find genes that may contribute to it, so they are more like susceptibility genes, which is a very relevant sort of thing to be thinking about when we think about autism spectrum disorders and other disorders. So where these two interact is where the disease phenotype can express itself, and the various kinds of things that seem to impact at the environmental level are toxins of various kinds. We're all carrying around body burdens of toxins that were never in our grandparents or our great-grandparents. And as Chris has just talked about aluminum, we live in the age of aluminum, so we're probably walking around with more body burden of aluminum than 100 years ago. Various other toxins such as various pesticides, various other heavy metals, changes in dietary things, viral infections, a whole list of things that can be actually changing the impact of various toxins in the environment, and of course various genetic factors, the various things that can cause the expression of the disease, those small fraction that are actually what are called the familial forms of the disease, versus a whole bag of things that seem to be susceptibility factors that we don't really understand. And I should stress that this whole area, this intersection, gene-toxin interactions, are not well understood at all. And in fact, they are almost unfundable by major granting agencies because when you try and talk about those sorts of things, you get back comments such as, this is an unfocused grant that wants to do too many things. And yet, realistically, that's where it's mostly going to shake out for most people. So, and again, susceptibility genes, some well-known ones. I haven't listed all of them. ApoE is a classic one for ALS-PDC and for Alzheimer's as well. And of course, that whole thing with age. Why is age such an important determinant? We know that ALS, Parkinson's, Lou Gehrig's are not, or Alzheimer's, are not diseases of young people as a rule. There are exceptions to that, but they are not largely diseases of the young. They are diseases of older age. Autism spectrum disorders is mostly a disease of younger age. So where are these things coming from, and how does age play a role? A whole bag of things I'm not going to talk about, but there are many speculations about what those kind of things can be. And what I haven't even mentioned is I haven't even talked about the impact of gender. Males and females get neurological diseases at different rates. And that's true for Alzheimer's, it's true for Parkinson's, it's true for ALS, it's also true for autism spectrum disorder. So how do we go from there to figure out what that collection of things that may cause neurological diseases might be? And whether you're looking at vaccines or whether you're looking at the broader world of things that could be leading to neurological deficits in Alzheimer's disease, There are thousands of environmental chemicals that are out there in the world. If you take the Merck index and open it up at random and start pointing pins at it, you will discover most things are neurotoxic if you try. You will also discover that that we're putting thousands more of toxic chemicals into our environment every year. And that, again, this is fairly well known. We also have something like 23,000 genes in the human genome. Any of them could interact in various ways as susceptibility factors to those toxins. So it becomes a huge number of possibilities you would have to explore if you wanted to look at all the possible toxins and all the possible synergistic interactions. And Lucia talked briefly about drug synergies and toxin synergies, whole unexplored area, and of course then the genetic component. So the only way we have thought to do this that seems to make sense to us is to seek out clusters. And a cluster, a disease cluster, is roughly defined as a large number of cases with constraints of time and geography. It's not just that someone on your block has ALS and someone 2 blocks over has ALS and therefore you have a cluster. No, it has to be a certain size and it has to be a certain space and time constraints. The best known example is a case of what was called domoic acid poisoning in mussels. Domoic acid is it's an agonist for what's called the kainate receptor, which is an excitatory amino acid receptor in various parts of the nervous system. And in Prince Edward Island in the late 1980s, there was a wave of people presenting to the hospital with acute gastrointestinal distress, in many cases followed by memory losses, seizures, and in some cases death. There were enough of them that people were able to isolate what had happened fairly quickly. The scientists were able to see that what it actually was people had been eating cultivated mussels. Cultivated mussels, which are filter feeders, had been accumulating phytoplankton as they will. The phytoplankton at this time of year had high levels of domoic acid. The domoic acid was going straight to the nervous system and starting to knock off cells in the nervous system. That's, again, a classic example of how clusters can work. But we don't find them very often in neurological disease. There are very few of them, actually. There are, again, that meet the standard criteria for a cluster. And for ALS, there are really only 2. One of them is this disease, ALS-Parkinsonism dementia complex of the Western Pacific, and that's why I focus my research on that. And the second one is a cohort of ALS associated with Gulf War syndrome. And this, when I'm looking for clusters, and that's really what I do and try and explore them, when I see a graph like this, which plots the U.S. Department of Education's rates of ASD, really we started looking in 1991, and Lucia will talk about this in much more detail. And you see a curve like that, which from my world describes what's called a power law function. That's something very disturbing, suggesting that something dramatic is happening. And again, Lucia we'll talk to you more about that. But it's not genetics, because the genetics of the population have not shifted, and it's not just better diagnosis. Again, I will leave most of that for Lucia. So let's talk about a little bit about what ALS-PDC actually is. ALS-PDC is the disease found in Guam, but elsewhere in the Western Pacific. The ALS component, which is a classical ALS with some little kind of weird proteinopathies that go along with it, was called lytico, from the Spanish paralitico, or paralyzed. And the other factor was a kind of Parkinsonism called Bodig. And Bodig looked like a non-classical Parkinsonism that had a very strong dementia component. So people who had Bodig, or PDC, what was later called PDC, seemed to have an atypical Parkinsonism, and some people actually got both. They got lytico-Bodig, and it was about 10% of the overall number of cases on Guam. Roughly the incidence in Guam was hundreds of times higher than anything comparable in North America. And that's really why it came to the attention of Leonard Kurland and other neuroepidemiologists at the time. This was in the years right after World War II. The American Army came back into Guam. They started to notice what seemed to be an extraordinary number of cases of ALS. And then as they looked further, an extraordinary number of cases of Parkinsonism. And they began to investigate this weird, what they called a geographical isolate. And even though there was clustering in families, and I won't show you family pedigrees, even though there was clustering in families, large numbers of siblings and families would have these weird collection of the features of that spectrum. Some would have ALS, some would have Parkinson's, some would have both, some would have nothing. There was no clear mutant gene identified, but a clear family clustering, some sort of weird interaction with genes for they code for ApoE, but again, no direct inheritance. A higher male than female ratio, which is largely true for ALS and Parkinson's, and certainly true for Alzheimer's up until menopause. And a very strange feature that we've now seen in Parkinson's disease as well, and as well as Alzheimer's, that one of the first kind of canaries in the coal mine was a disturbance of the olfactory system. People just don't smell things as well. And actually, that was actually demonstrated up at UBC long before my university, long before I became involved in the study with a series of MRI studies. So what was it? Epidemiological studies were conducted. A ethnobotanist named Margaret Whiting came out to Guam on the invitation of Leonard Kurland and basically started looking around. Guam, again, when you think about the 23,000 human genes, and of course we used to think there were more, but 23,000 human genes, the thousands of potential toxins, and Guam was a toxic environment after World War II, it's a big haystack. But it's a smaller haystack than looking in North America. So there's a needle in there somewhere with this disease. The very clear disease cluster. Could it be? Margaret Whiting looked at everything. She looked at radiation. She looked at military toxins that were spread all over the island. She looked at any number of things that people were eating. And what she finally decided, after detailed epidemiology, is that it really was tied to a toxin contained in the seeds of this plant, the cycad. And again, we won't go into much detail about that. This is what the seeds look like, and the people have historically been using these seeds as a dietary staple in times of famine for hundreds of years. This is what they look like when you take them apart, the gametophytes, and cut them up, and they make tortillas and dumplings and things like that out of them. The peak of the ALS-PDC epidemic was in the 1950s and 60s, again, which hit something like 25% of the adult population died from some combination of that spectrum, and it has since declined. Something has changed on Guam. The disease that used to be at a very, very high level has now fallen to approximately North American levels for the ALS component. The Parkinson is still a little bit higher, but it is now largely within the boundaries of what the North American experience. Having said that, the North American incidence level has gone up. Related islands, the same Chamorro people indigenously, Saipan occupied by the Japanese, cut down all the cycad trees during the 1920s. The Chamorros in Saipan ate no cycad. They had no ALS-PDC. The Guamanians ate cycad throughout the war because they were in a famine diet, and they ate high concentrations of cycad and had that explosion of that disease. Again, it remains the single largest cluster for ALS, Parkinson's, and Alzheimer's in the world. There is one new exception of Parkinsonism found on the island of Guadeloupe in the French Antilles, where people consume another fruit that has high levels of something called nonosins, which are a neurotoxin as well. A whole series of possible toxic compounds found in cycad, and I'm not going to go into those due to lack of time. And this is just some of them. We think we know what some of those toxins are, but we'll save that for cocktail discussion. But one of the factors that caught my attention at the time was a Nobel Prize winner named Carleton Gajdusek went there in the 1980s. And Gajdusek, I think arguably is a fairly smart fellow, and he was convinced that it was high aluminum in the soil. And he actually looked and measured the soil across the island and found that in the southern parts of the island, where they actually had ALS-PDC hotspots, that there was an awful lot of aluminum soil, abnormally high. And it had something to do with, and Chris can correct me on this, had something to do with volcanic activity and aluminum shifting around through the Earth's crust. But in any case, a lot of aluminum there. He was convinced that that was the factor. Others disputed that. That was never really followed up. So when we got involved in this, without knowing what the toxins were, because again, there was a whole Christmas stocking full of potential toxins, we said, let's just do a really stupid, simple experiment. And we'll come back to why this, I think, is a good way to go. Let's just remember, the epidemiological validity was, take the cycad that the people are eating, that is linked to the disease. And now we are going to see if we simply take regular old colony mice, and we'll make them just to keep it simple, they'll make them all boys and make them all of a certain age, and we'll feed them the stuff at about the ratio that people had calculated people were eating. In other words, about 25% of the diet. We're just going to feed them cycad seeds. And we had, of course, analyzed the cycad. We knew how much of all this stuff was in any batch of cycad. We're going to feed them cycad. What's going to happen? And moreover, we're going to look across what we call 4 dimensions. We're going to say, what happens behaviorally? In other words, what are the changes in motor functions? Because we're looking for an ALS-like phenotype if it exists. We're looking for those cognitive and olfactory changes that might also occur. We're going to look at a cellular level, what's happening to the cells in those parts of the brain and other parts of the brain, what's happening at a biochemical level, and crucially, we're going to do it across time. It's not just going to be a snapshot, but we're going to look at as many time points as we can conceivably do. And you can imagine this logistically turns into a very large experiment with lots of mice very quickly. So some of the motor tests we've done, really simple ones. We have much more complex ones now. This is animals moving on a turning wheel. It's called a rotorod. Standard test in ALS research. Something called wire hang. You measure muscle strength, put a mouse on a grid and just invert it. How long does it take them to fall off? This is a standard test for what's called alpha motor neuron integrity. A mouse, if you lift them up by their tail, will splay out their hind limbs. And that tests the alpha motor neurons in the spinal cord, the ones that are in fact lost in ALS. And you can actually measure across time the impact of that leg extension function or dysfunction as you feed them cycad. They begin to lose that function. And that loss of function, I should also mention, a Parkinson's feature, many people will know that as people with Parkinson's progress in the disease, the gait becomes very disturbed. How they walk is a classic feature of Parkinsonism. And you can do that with your mice as well. And again, much more complicated ways. You can also do fairly sophisticated imaging, and you take the brains and the spinal cords of these animals and have them analyzed ex vivo for structure. And this is an example of magnetic resonance, what's called microscopy, because it's beyond imaging. The resolution is so great that you can almost see cellular structures. With the magnet we're using with our colleagues in Florida, we have something like a 40 micron resolution. So that's pretty remarkable. And you can then use volumetrics to actually calculate the areas, the volumes and areas of those different brain regions. And again, this is just an example how you can actually look at different things, whether it's hippocampus, substantia nigra, other areas involved in ALS-PDC. And here, of course, the spinal cord, the areas of the spinal cord that are affected in ALS-PDC. And for example, with those volume reduction programs, it's fairly easy to calculate and demonstrate that, for example, in the ventral horn, the area that's primarily affected in ALS, the lumbar spinal cord, you will actually lose something like 20 or more % of the overall volume. There's that graph I showed you again. Here's your volumetric analysis. And you can count motor neurons. How many motor neurons are there? How many have survived? And in our mice, over a period of time at which we usually ended the experiment, we had lost something like half of the motor neurons that were in that part of the spinal cord. I'm not going to show that because I've got a better picture of it. And this is a very, very busy slide that pretty much is there to remind me that this is, after all, a model of ALS-PDC. And those different regions, the same way the ALS component happens in humans, a loss of motor function, a loss of motor neurons, a loss of volume, and very, very unhappy, sick-looking motor neurons found in the spinal cord, and a loss of various biochemical markers of things that should be there. The area that's affected in Parkinson's disease, called the striatum substantia nigra, also at the behavioral level, they don't walk the same way. There's a loss of volume. The cells are showing what's called programmed cell death, and a loss of a critical molecule that's involved in the synthesis of tyrosine, of dopamine, called tyrosine hydroxylase, a standard marker for dysfunction in Parkinson's disease. You look in the cortex, again, a memory function. The animals are less capable of doing it. Cortical thickness is smaller. Number of the volume of the hippocampus, the region involved in short-term memory, is smaller. Areas of hippocampus and cortex show a loss of neurons by this programmed cell death. And again, this kind of weird, patchy mislabeling, and Russell, this may be relevant to your stuff because these are glutamate transporter antibodies, and you show this weird, patchy distribution of the transporter, suggesting things have actually fallen out of various kinds of cells. And when you compare across between our mice and the actual disease, once we had done that first comprehensive study, and I should cite a reference, of course, so I'm making everyone else do. This is Wilson et al. And a series of subsequent studies that we published since 1972. The number of features in the human disease and that we see in the mice looks remarkably similar. And again, mice are mice and people are people, and we know that. But that the behavioral dysfunctions, the loss of neurons in different neural populations, and the kinds of things that are happening, such as a change in dopamine receptors and dopamine transporters, they're all quite similar across the different conditions. So are there any similarities between ALS-PDC and Gulf War syndrome ALS cohort? And the answer, we think, may be yes. Again, we had used CyCAD as the way to demonstrate the toxicity. We also isolated the molecule we think is the toxin. We've used that, and I'm not showing you those data, which also give most of the same features. But we've never really come back to look again at the whole issue of aluminum. And aluminum, again, in those areas that are hotspots for ALS-PDC is extremely high. And one of our colleagues, Tom Marler, who's a botanist in Guam, is now actually doing that. He's going back to the CyCAD seeds and doing the soil and water samples, again, in addition to the plant samples, to see if they have unusually high levels of aluminum. Gulf War syndrome ALS. Many things were suspected. Again, we searched for clusters. This is the second cluster of ALS. When we heard about it, we said we're going to follow it up. But what could it be? The Gulf War environment, the theater of operations for the military of any country, was an extremely filthy environment. There was depleted uranium. There were all kinds of munitions. Of course, the soldiers were under stress. They had numerous chemicals they were taking as protectants against various things such as nerve gas, and they were also getting massively vaccinated. And the one that most caught our attention, the one that the epidemiology seemed to suggest might be linked, there were basically two things. There was an anti-nerve gas agent that they were taking in pill form, and then there was the aluminum in the AVA vaccine. And that's when we first started talking to Merrill Nass and others, and we started to wonder if, in fact, the aluminum or anything in the AVA vaccine could be a cause of the loss of motor neurons. How am I doing for time?
Minutes.
Perfect. And this is where I found myself in my career going down the rabbit hole, because we started to look for ways to actually study the AVA vaccine. And without going into a lot of detail, found that we really couldn't do that, but we could pull the vaccine apart. We couldn't get the PA, protective antigen, because they wouldn't release it, but we could from the company at the time. But we could say, okay, what do we know that's in it from the product safety sheets? And I'm just showing you another product safety sheet as an illustration of what kind of the general problems are. But for example, there was aluminum hydroxide in fairly high concentrations. There was a suspicion that has been denied that there was squalene, and there are possibilities, of course, of all the other kinds of things that go into vaccines as preservatives and as other bits and pieces that Lucia talked about the other day that might be part of the problem. The H1N1 vaccine that came out last year was adjuvanted somewhat differently. It had something called ASO3, which is a squalene-based adjuvant. But to the best of my knowledge, squalene has not been well tested. It did not apparently have aluminum, and I'll come back to why I am not entirely convinced that that's true. And it said things that this was totally safe. So I've highlighted a piece from there, the product safety sheets that came out last year. And again, I have them if anyone wants to see them. They actually make for some interesting reading. Of course, they say non-clinical data revealing no special hazards for humans. Okay. And then they go on to say where they had done a study on pregnant rats, and they had injected the adjuvanted and non-adjuvanted forms. And in the adjuvanted forms, they had these sorts of things: fetal malformations, medially thickened kinked ribs, bent scapula, increased incidence of urulated ureter, and the one that caught my attention, delayed neurobehavioral maturation, which of course made me think of the kinds of things that Andy Wakefield talked about yesterday. They did the study again and said, oh, well, we didn't see it the second time, so clearly means nothing happened. And hence, toxicological significance is uncertain. And sadly, when we first began to get into this literature, we started to look at the various kinds of company reports that were out there for any of the adjuvanted vaccines. There's one from Chiron that it's sort of problematic for a number of reasons, where you could not actually draw a conclusion based on what was in the product safety sheets because of the number of animals that they used, or the level of analysis they used. So for example, the Chiron Corporation, and I don't have any reason to believe these guys are a whole bunch better, Chiron Corporation basically looked at their animals and they said, they look okay. There was no behavioral analysis. And as we saw from the stuff on ALS-PDC, it takes a bit more experimental scrutiny to actually demonstrate the deficits in behavior in these animals. Because remember, in neurological disease, you don't show the neurological deficits until you've lost 70% of those cells in that part of the nervous system, or they're dysfunctional. So when you see a statement saying that the mice look okay, that's not quite enough. Chiron study did no histology, as far as I know. This study, as far as I know, doesn't report any histology. So I don't actually know what the nervous system of these animals looks like. Aluminum adjuvants are used in a whole series of licensed vaccines. Again, I'm not going to go into it because Lucia will. But it is a fairly common, various of the aluminum salts, the hydroxide, the phosphate, and the sulfate are widely used. What the percentage is, again, I'm not sure. Maybe Vicky can inform us, but it's high. A lot of vaccines are adjuvanted with aluminum. So how did they get in there? Why do we have aluminum in vaccines at all? You actually have to go way back in time, and back to the first paper I could find by Glennie and colleagues. It came out in 1926. They were working for, I believe, Burroughs Wellcome, and they were having some issues because their vaccines were not producing consistent antibody responses after a certain time. So they literally, and again, you have to read the paper, it's entertaining in its own right, they literally started to go through the chemical shelves and tried various things. And they have a series of papers. They try various things. They try phenol, they try all kinds of stuff, adding it to their vaccine preparation, and they stumbled across alum. And I don't know what the logic was of that, but aluminum in various ways had been used in canning, it had been used in dye fixation. It had been well used for over 100 years at that point. I'm assuming they thought it cannot possibly, if it's in your pickles, it cannot do you any harm, and therefore if we put it into your vaccine, it can't do you any harm either. And the rabbits, or I think they were using rabbits at the time, demonstrated a persistent antibody response. And thought, we're there. We have now found the adjuvant, the helper that will make the vaccine actually work. And that's in contrast to scroll forward 80 years or so when a conference was held in Puerto Rico, reported in Vaccine, conference report summary, 10 things we know for sure. First thing is that vaccines have saved aluminum adjuvanted vaccines have saved millions of lives over the years. Okay, let's say that's true. Then scroll to the end of the paper. Here are the 10 things we don't know. We don't know anything about the toxicopharmacology of aluminum in infants, small children, young children, and pregnant women. Oh, wait a minute, that's a lot of treatment groups. So I don't know how that paper could have been written that had those two very divergent sets of conclusions. So is aluminum toxic? Chris has shown you that the aluminum certainly can be toxic under various circumstances. I'll show you some of our data in vivo and in vitro. In a cell culture dish, this is our aluminum hydroxide, and again, we'll come to the in vivo experiments in a moment. These are what are called NSC34 cells. These are hybrid cells that are derived from mouse motor neurons and a neuroblastoma and merged together to make this kind of cells that had motor neuron-like properties, which is the reason we use them. As you raise your aluminum hydroxide concentration in cell culture media, you kill more and more cells. This is just a percentage cell count. A little bit better count is with a different kind of cells called IMR32 cells, a fibroblast-derived human cell line, very susceptible to aluminum. This is a aluminum chloride, actually. We're doing both experiments with both cell types. Aluminum chloride, in a very short time, it kills, and this is an MTT assay, a cell viability assay, kills an awful lot of cells fairly quickly. What does it do if you actually give it to animals? Because as I think we all know, cell culture stuff as a model system has its applicability, but cell culture is also very problematic. Those of you who've worked with it know you can look at your culture funny one day and the cells will die. So you have to take that with a grain of salt. And it's also an extremely artificial environment. But what about if you take aluminum hydroxide and simply do that four-dimensional experiment in mice? The same way we did with our cycad feeding and our other toxin exposure in the mice, the same way we were trying to duplicate ALS-PDC. What if you simply do that four-dimensional model in mice? And this is the Petrick et al. Study that came out in 2007. If you look at muscle strength measured by the wire mesh hang, they lose that very rapidly. And this is what these mice have had, and we were trying to duplicate as closely as we could the conditions of American soldiers who had received the AVA vaccine. There's a lot of discrepancy in the literature about whether they received 1 shot, 2 shots, 3 shots, 6 shots. No one knew, okay? And so we said, it sounds like the bare minimum is 2. So we'll do 2. We will scale it down to a mouse. The mice will get the equivalent body burden of aluminum hydroxide injected subcutaneously, same way that the AVA is administered. These are young mice, young male mice, to keep it really simple. So we control the age. These are colony outbred CD1 mice, so there's no weird genetic background to them. And all we're going to do is we're going to watch them behaviorally for a period of time, sacrifice them, and start looking inside the nervous system and interrogating the nervous system to see what might have changed. So wire mesh hang, significant loss of muscle strength fairly rapidly after the second shot, which is right here. Okay. Changes in their open field behavior, how they wander around this kind of a sawdust arena. And at the late stages, at least with some of the compounds, aluminum and squalene, they actually, because we combined the two, they showed a loss of ability to remember where they were in that water maze. At a cellular level, you can actually go in and use a molecule called choline acetyltransferase, which is specific for acetylcholine-containing motor neurons in the spinal cord, and you just go count them. And what you see is when you do that, that the aluminum hydroxide-treated animals have roughly a 40% loss of motor neurons in the lumbar spinal cord. And another example of that, here are the control animals. Here are the aluminum-treated animals. This is something called NVIC, which labels neurons. And this red is actually a stain for programmed cell death. Okay, it's called caspase 3. And what you actually find out is that there are fewer motor neurons, as we've already seen in the graph, but they're actually co-labeling. The surviving ones are actually co-labeling, where you see the yellow. That's where you've actually merged this color with that color, and the yellow is where they're expressing both. So these are neurons, they're motor neurons, and they are showing the signs of programmed cell death. And here's a good blow-up. This is just the caspase itself. Here's the merged image in a motor neuron that seems to be undergoing cell death. Different regions of the brain have, and the motor system have different impacts. Very, very strong impact in the lumbar cord, where ALS often begins. Motor cortex, also a site for ALS, is also impacted, and lesser effects elsewhere. So it was, in this particular administration, a very profound impact on the motor system. You can look at what's called glial fibrillary acidic protein. It's GFAP for short. It marks a kind of cell called an astrocyte, and particularly a kind of astrocyte that shows up at the site of injury. It goes up very dramatically. Here's what one looks like at a higher power. If you stain the cells for what's called the Morin stain, you can actually find that the motor neurons, in many cases, are apparently full of aluminum, and only the aluminum-injected animals have that. Again, GFAP, you can see, is massively elevated. EBA1 is a marker for reactive microglia, vastly elevated in the aluminum-treated animals. So you have the signs of what's called neuroinflammation. The glial markers are all up. And we even see something that's been reported in the literature before associated with aluminum, that in the aluminum animals only, this is a slide taken from the cortex of someone who has ALS-PDC. This is a slide stained for the hyperphosphorylated tau protein that forms the neurofibrillary tangles that are a classical hallmark of Alzheimer's disease and ALS-PDC. There's a very clear tau-labeled cell, and when you come down to the spinal cord of the aluminum-treated mice, you also get very clear tau labeling with the same kinds of little pathological inclusions that seem to be very, very similar. Second study done by Petrick, reported in Shaw and Petrick 2009 in Journal of Inorganic Biochemistry, a whole series of biochemical changes, behavioral changes that seem to happen in aluminum-treated treated mice. These are animals injected a little bit older, a few more injections, but the same general idea. So for example, how they walk, their velocity changes, a whole series of things. I don't have time to go into all of them. By the way, these are all statistically significant. When I transferred the files from the paper into the PowerPoint talk, a lot of the significant stars didn't transfer, but if any of you want to see those data, I have them on my PowerPoint. Many things are changing in how their motor function works, and that becomes still a very, very profound impact on the behavior of the animals. In the water maze, how they actually remember where it's a visual, it's a test of visual memory, basically, spatial memory. And the animals that have aluminum take significantly longer to learn that task. And the significance value is quite high. This is a more recent study, really hot off the press. And again, these are all highly significant values. We have, with a new way of analyzing motor behavioral function, we have something called a digigate system that actually measures 32 movement variables. And it's a very sophisticated camera, computerized system. And mice run down this little track, and you can actually measure exactly how they place their paws and how long their stride length is and how they turn their paws. Many things you can do with this device. And in many of these areas, their functions have changed very dramatically. These are data that come from an open field test. One thing that jumps out is that the animals that are treated with aluminum, they don't like being in the center of the open field. They hang to the walls, and that's a phenomenon called thigmotaxis, and it's usually a function of learning disorders and/or anxiety. So the animals will spend less time, more time at the borders. They'll go less into the open area, and their latency actually to cross into the open field is extremely delayed. The animals just don't like doing it. Again, classic sign of some sort of disorder. And again, coming back to those pesky ASD data again, what are we to make of that? Lucia, how am I doing?
5 minutes left.
Lucia, again, we'll talk about this in more detail, but what we will show, and I believe, and look forward to your critique on this, is that when you look at aluminum adjuvanted vaccines and look at the years in which those rates have changed, that you see actually there's a huge spike in percentage of cases that starts in about 1991, and that if you plot the US Department of Education ASD data against the CDC's vaccine schedule year by year, you get a very, very tight correlation coefficient that turns out to be significant at the 0.0001 level. And again, we recognize this is correlational and it's not causal, but it's still interesting. And I would simply suggest that if we had a 0.85 correlation coefficient and I told you it was ice cream, NIH would be a lot more interested in finding out what flavor of ice cream might be involved. Whether or not they will be as interested when we talk about vaccines and aluminum, I don't know. So conclusions, what we've done so far, and I haven't been able to, in the time allotted, to give you all of the things we've done, but I think we've shown that there is certainly evidence for aluminum in some neurological disease states, certainly in an animal model of, and again, with the caveats that go with animal models, that some of those impacts involve motor neuron loss and neuroinflammatory changes as well. There are very clear behavioral deficits of both motor and cognitive function. But we need to do a lot more. We are nowhere near done with what we need to do. We need to understand the different kinds of aluminum compounds. And again, Chris touched on that. There are many kinds of aluminum compounds out there. They come at us and at our experimental animals by different routes of administration. There's an issue of dose. There's an issue of duration. Crucial issue of age. Is dietary aluminum or injectable aluminum as much of a problem if I get it versus a newborn baby? I think so, but that has to be demonstrated experimentally. There's clearly an impact of gender, and that whole area of gene-toxin interactions is something that remains largely unexplored. People will often say, well, if ASD is now roughly 1% of the children in North America, 99% are fine, so it clearly can't be that. Well, that's a little bit like the argument of Guam. So Guam, 25% of the adult population had ALS-PDC, 75% didn't. that's where the gene environment stuff becomes interesting, because something that 1% are doing or not doing may be the reason that they become more susceptible. And it then becomes partially a discussion of concentration, because as you push the concentration higher, you can, if that's the causal factor, you can expect that population to shift to higher levels yet again. So we definitely need to look at gene toxin things. We need to look at key biomarkers. Oxidant stress, for example. Is that happening in different areas of the nervous system? Can we predict where it's going to be? We need to look at stress markers. And frankly, we need to actually go back to some of the vaccines and say, what's really in them? If we can't necessarily trust the product data sheets for amounts, or correction, for the rigor of the analysis of the impacts on animals, are we really sure how much stuff is in batch to batch? And I think there's no way to find out but to go do it. And ultimately what we really need to do, because whether we're talking about ALS or Parkinson's or Lou Gehrig's disease, and the possibility that aluminum can have any role in any of those diseases, let alone the impact on children with autism spectrum disorders, what are we going to do about that? How are we going to, it's one thing to say, okay, if that's the cause, prophylaxis, just don't do it anymore. Sure. But what about all the people that are now in those conditions? How do we help them? How do we start to remediate the damage that may have been done by aluminum? So a lot of people to thank. Various granting agencies have funded the work over the years. Most of the stuff I've told you. These are the grad students in the lab. Jason Wilson being a key one who had a major role in developing the ALS-PDC model, and Lucia, who has now come to my laboratory as a postdoc and will be working with us on kind of a much more elaborate discussion and an investigation of aluminum toxicity in various kinds, and various colleagues elsewhere. And of course, very much gratitude to the Dwoskin Foundation and the Caitlin Fox Foundation for providing us with the funds to begin that next stage of our work. Thank you very much.